Well, there are a number of problems with the RNA world, and I'm happy to speak to them all in the question and answer time. I have an extensive chapter about this in the book. It's interesting to note, for example, that Christian de Duve and other people who are very much in the forefront of origin of life studies are now saying that we now need a pre-RNA world because the RNA world is so fraught with difficulty. But for the purposes of my talk, I want to just highlight one of those difficulties, and it is again an information problem. The RNA world envisions an RNA molecule capable of copying itself. The backstory on that is that they're called ribozyme engineers. Scientists are working very hard to try to get RNA to do the things that we hope it would do if the RNA world hypothesis were to be true. Have found that in the best of cases, they've been able to get an RNA molecule that can copy about 1/10 of itself, about 1/10 of the bases. But to do that, even to get that limited replicase capacity, requires a huge amount of information in the form of very precisely sequenced bases that have been sequenced by an intelligent agent, namely the ribozyme engineer. So what exactly does that simulate? Well, some might say it simulates the role of intelligent design, but setting that point aside, it certainly shows that the RNA world does not account for the origin of information. Again, it too presupposes information just to get that primitive molecular replicator going. So this whole approach of relying on natural selection, whether with simple organisms or simpler molecules, has run into a very imposing obstacle. Now, the third approach recommended by Monod's categories is the approach of necessity or law, or what goes by the name in origin of life studies as self-organizational scenarios. The idea here was very simple. It was proposed first in the late 60s and early 70s, and it was the idea that perhaps these large biomolecules could be likened to something like a crystal, where the forces of attraction between the constituent parts determine the order, the sequencing of the chemical constituents of the molecule. A crystal of salt has two ions, the sodium with the plus charge and the Cl with the minus. There's a force of attraction. That force of attraction generates, in a very self-organizational way, a beautiful, orderly crystal lattice. Two scientists, Kenman and Steinman, proposed that perhaps that's what's going on to explain the origin of the information, the sequence specificity in proteins. Maybe that's what's at work in nucleic acids as well. Now, they after several years working on this proposal, they wrote a very important book called Biochemical Predestination. They weren't Calvinist theologians, they were chemists, saying that the structure of DNA, the information it contained, was predetermined or biochemically predestined by these forces of attraction between its constituent parts. Now, they ended up repudiating their own theory. They had invoked this initially to account for the sequential arrangement of the amino acids in proteins. They found that there were some slight differences in the bonding affinities between the amino acids, but none of them correlated to the arrangement of amino acids in known proteins. But with the DNA, they found that there was an even deeper problem. It was Kenyon who overtly repudiated this idea. And the problem was this, and I need to direct your attention to, good, it works. The structural formula for the DNA molecule. The pentagons and the sugars form what's called the backbone, and there's one on each side. The A's, C's, T's, and G's in these little boxes represent the nucleotide bases. Remember that it's the sequential arrangement of those bases that conveys the assembly instructions for building the proteins, for arranging the amino acids in the right way. Now, what they were trying to do was to explain the sequencing, why the arrangements are just the way they are, by reference to forces of chemical attraction. Now, notice that in the molecule, there are these, in the way we represent the structure, there are these little sticks. The sticks represent chemical bonds. Now, notice that there are chemical bonds between the sugars and the phosphates, chemical bonds between the sugars and the bases, but there are no chemical bonds between the individual nucleotide bases. Now, this has a really powerful implication for their self-organizational model, because If there's no chemical interaction here, you can't invoke chemistry to explain the sequencing. You can't even invoke the attachment site here because this bond is called an N-glycosidic bond for people who are keeping track on their scorecards. This bond is the same in every case, so that at each site along the DNA chain, any one of the 4 bases can attach. It's chemically indeterminate. Any one of those bases can accept, or any one of those sites can accept any one of the bases. The chemistry, in other words, is not determining what attaches or the arrangement of the bases that attach to the medium. The message is here, the chemical message is here, the medium is here, but the chemistry is not determining the sequencing. When Kenyon realized this, he abandoned his whole self-organizational approach. I have a visual illustration, especially for people who aren't chemists. Forgive me. In case this structural formula of DNA didn't make a lot of sense. I've got a little message up here that combines some scientific terminology with American slang. The double helix rocks. All my students used to use rocks as a verb to say they liked something. Now, what we have here is a physical analogy to what's going on in the DNA molecule. In the DNA molecule, you have the backbone, the sugar phosphate backbone. It's not the message, but the medium upon which the instructions are inscribed. I have a medium as well, a backboard, a chemical or a magnetic chalkboard. The little tiny message I have here is also stuck to the backbone with a force of attraction. It's a magnetic force of attraction. But notice that that magnetic force of attraction does not determine the arrangement of the characters in this little informational message. That's determined, well, Michael Polanyi, who first recognized this fact of DNA, said that the information was extrinsic to the physics and chemistry. It's coming from outside the physics and chemistry. Note, we can tell that the magnetism is not responsible for the information because I can quickly rearrange things. We have all the same magnetic forces in play, but we have destroyed the specified information that was there originally. So this is what this helps, I think, understand what's going on in DNA. It's not the physics and chemistry that's determining the messaging. Moreover, if it were the physics and chemistry, it turns out that what you would end up with is not an information-rich sequence, but instead something that information theorists refer to as redundancy, or simple repetitive order. If every time in that DNA molecule you got a T, you turned up a G along the spine, then what you'd end up with is a kind of repeating messages of TG, 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 TG. You'd get a molecular mantra, not a message, not an information-rich sequence or that could direct protein synthesis. So both for empirical reasons and in principle theoretical reasons, scientists working on this self-organizational approach abandoned it. If it had been the case that self-organizing properties were responsible for the information in DNA, we would have been actually incorrect that it was informational.